So yeah, I was saying that sleep is essential. We would agree, and we sleep a third of our lives. And sleep-like states are common across the animal kingdom, be it mammals, birds, flies, worms, fish, reptiles. So although sleep might look different in different animals in terms of duration, in terms of the posture of sleep, but they all sleep. And at least in all the studied animals, sleep or some sort of sleep-like states have been found. Another way to assess the importance of sleep is to look at the impacts of sleep deprivation, which we would agree they are adverse on memory or learning or efficiency of working and overall health, right? So if we drive when we are sleep deprived, it's highly probable that we'll meet with an accident. So overall, sleep is very essential, and sleep disorders like insomnia and sleep apnea are very common across the world, and abnormal sleep is a comorbidity in many psychiatric diseases like Alzheimer's and autism spectrum disorder. Even then, the mechanisms underlying sleep are not very well known. No one actually knows what's the function of sleep. There are theories. Why is there a downtime in our life? Why do we need to turn down for 8 hours in a day? So, there's still an active research going on to find out what are the mechanisms that regulate sleep, like why do we at all fall asleep. So now there is a sleep is not a homogeneous or a uniform behavioral state. It's made up of multiple substates. At least in mammalian sleep, that has been shown over and over again, and each of these sleep states can be measured and studied separately. For example, these are few traces from human EEG showing you 4 different stages of non-REM, non-rapid eye movement sleep, and REM sleep and awake state. So each of them can be distinguished from each other based on the frequency component, the amplitude, and the synchronization between different channels of EEG. So, and more importantly, each of these stages are thought to be linked to different role in the brain. So, for some of them are thought to be linked to help the brain in memory consolidation. Some of them are thought to link to maintain like body homeostasis, brain homeostasis. So, they are thought to have different roles in our brain. So, and our brain over a duration of time, let's say over 8 or 10 hours we sleep, it alternates between all these 4 stages of non-REM and REM and wake stages multiple times throughout the night. So it looks like all of these states are important in some way or the other. Now this was all about mammalian sleep, and now there is emerging evidence that several species of birds, reptiles and flies also have different states of sleep, which means this might be an evolutionarily conserved behavior. But to what extent is this largely true across vertebrates is not very well known, and it's an active field of research. Now coming to the zebrafish, which is our hero in the talk today. So zebrafish, so it's like this is like a 4-day old zebrafish post fertilization. It is just 3 to 4 millimeters long at this age, and it satisfies the behavioral criteria for sleep, and I'll get to those criteria in a minute. But apart from that, it offers several unique advantages to studying sleep. For example, it's a vertebrate, we all are vertebrates, and it has diurnal sleep patterns, which means it sleeps during the day, sleeps during the night and it's awake during the day. It's anatomically and molecularly similar to mammalian brains, which means that the findings that we make on zebrafish brain could be translated to our sleep. And its brain is transparent actually, right? So it's because of transparency and small brain size, it's well suited for whole brain imaging. So that's one of the model organisms in which you can record the brain activity from all the around 10 raised to power 5 number of neurons simultaneously. So that's a huge thing, and it's amenable to genetic, chemogenetic and pharmacological approaches because of lack of matured blood-brain barrier, which means that the scientists are able to genetically manipulate and induce certain sleep disorders, sleep deficits through genetic perturbations, through chemicals, and through like just adding drugs to the water they live in. So these are several advantages that it offers, so which means that it is a very well suited animal model to understand sleep. And now I'll just quickly go over some of the methods that we used in zebrafish sleep research. So one of them is movement tracking. So what is done is, so these are like offspring of two zebrafish. So when they are 5 days old, that's when their sleep cycles mature, which means they have consolidated sleeps during night time and less sleep during the daytime. The larvae, single larvae, are put in the wells of this plate. So there are 96 wells in this square plate, and then they are imaged or video tracked for over 2 days, and then their activity is recorded, like during day, during night, and you can see that during day they have higher amplitudes of activity as compared to during night. So I'll get back to this behavioral recording in a minute. Then the second thing is whole brain imaging, like I told you, and this is a name of the microscope that one of our collaborators have developed. This is 2-photon selective plane illumination microscopy. So that allows simultaneous brain imaging. So this is a brain of the larval zebrafish. So this is the top view and this is the side view, and what you are seeing here flashing are neurons that are getting active or inactive, and this is just a single snapshot. And there is a simultaneous behavioral recording, which means that the tail movement of the fish is also recorded. So this is simultaneous behavior plus brain activity recording in this setup. And then there is a component of computational neuroscience, which right now what I will be talking about is model fitting and model development, where what you see here are the states 1, 2, 3, 4 states as a function of time. So few of these states are taken by the fish during the night, and rest of them are taken during the day. So this is known as hypnogram, sleep hypnogram in normal terms. And this is a schematic of two process model of sleep. So this was given by a scientist known as A. Borbely for human sleep, and he proposed that sleep is staged by a complex interaction of homeostatic process, which is shown here by H black curve, and circadian process, which is shown here by these blue and red curves. So he proposed that sleep pressure develops during the day and drops during the night, develops during the day, drops during the night, and the thresholds at which it drops and begins to rise up again is determined by these circadian components. But I'll not go into the detail of this, but I'll quite a lot talk about this part. And then there is finding the neural correlates of sleep and wake. So once you have activities of each of the neurons versus time, you can correlate it with sleep and wake, like which neurons fall asleep and which neurons correlate with wake, which means that they are active when the person is active, they are active when the person is sleepy. So finding neural correlates of sleep and wake, and the last thing is sleep perturbation experiments. So sleep perturbation is a very important aspect of assessing sleep, because you need to perturb it somehow to measure it, right? You need to wake up the sleeping fish by sleep depriving it, by giving it some tapping stimulus, or you want to make the waking fish to sleep by adding melatonin in the water it is kept. So sleep perturbation experiments and analysis. So this is like a few of the methods that current research in zebrafish sleep people are doing.
Question.
Yep.
Zebrafish, so sleeps around 10 to 11 hours.
No, so this is like a general schematic. This is true for human sleep, but we are taking some motivations from what is known about mammalian sleep to then understand zebrafish sleep.
So how long do they sleep?
So it's very tricky. So it's not consolidated like us that we sleep for 8 hours. Their sleep duration is 1 minute, and 1 minute of inactive duration is counted as sleep in larval zebrafish. But that durations is more frequent during the night as compared to day. So they have more number of sleep bouts during the night as compared to day. Yeah.
So then how does one distinguish just from when they're just not?
Yeah, that's a very good question. So, like separating quiet wake from sleep is a very interesting problem, and one has to first define what is sleep. So, what is done is like there have been experiments which are known as arousal threshold experiments. So, zebrafish who are quiet, they are disturbed, and then how much time they take to respond to show some activity is plotted as a function of pre-stimulus rest, and around 1 minute there is a huge jump in response latency. So 1 minute is counted as a sleep, like there's an onset of sleep now. Yep.
As they go in the adult stage,
Yeah.
the sleeping duration.
That's a very good question. So in adult zebrafish, the sleep duration is 5 minutes. So if they are inactive for 5 minutes, that means they have entered sleep now. They are not like quietly awake, they are sleeping. Yeah. But larval zebrafish, as I told you, it offers several unique advantages like brain imaging. Their brain is transparent and it's small, so you can do microscopy and stuff as compared to adult zebrafish. Yeah.
So in the 1 minute, I assume they're gonna in night they have more minutes spending in
Yes, yes. Now, the question that I'll be addressing today is, can we identify distinct sleep states in fish just based on behavioral data? Now, what I've shown you earlier for humans was brain data, right? The EEG was brain data. But I'm asking this question just based on behavioral data because doing long-term electrophysiological experiment, while non-invasive experiments, is next to impossible in zebrafish right now at least, but people are doing it, but then the statistical power is not good. We cannot do it on 100 animals, the EPhys, electrophysiology. So we begin by asking this question on behavioral data, but our ultimate aim is to go inside the brain and see what these sleep states look like inside the brain. So precisely, I'll be talking about what are the number of sleep states in larval zebrafish, what are the differences between these sleep states, and how are these states altered by genetic and pharmacological perturbations. And now I must say that this work is supported by a UO1 grant that is shared between 3 labs, the Probal lab at Caltech, who are experts in studying behavioral, anatomical and molecular mechanisms regulating sleep in larval zebrafish, the Truong lab in University of Southern California, who are experts in developing this 2-photon microscopy like I just told you, and the Goodhill lab where I am at WashU, who are experts in developing computational methods and generating predictive mathematical models. Right. Now, I was telling you about the behavioral criteria of sleep. So, when do we call a person or anything as any species as asleep? So, sleep primarily occurs during specific periods of time. So, you are more sleepy at a particular time during the day as compared to others. And then the next is there's an increased arousal threshold during sleep, which means that it's hard to wake up the person when they are sleeping versus when they are like quietly resting. And it's even harder to wake them up when they are in deep sleep as compared to lightly sleeping. So this is the second criteria, and the third is the sleep is controlled by homeostatic system, which means following sleep deprivation there is a need for sleep. You are more sleepy after you are sleep deprived, right? So we'll keep these criteria in mind whenever we say, okay, in zebrafish this is known as sleep, or these are sleep states. So we'll ask whether these 3 criteria are satisfied or not. Right. Yeah, like homeostasis is like maintaining either the temperature or anything in the brain, maintaining its state so that it can function normally. So when the person is sleep deprived, there are certain deficits in the brain that keep developing, that keep worsening. So sleep is needed to get them back to their normal state. So when you're sleep deprived, there is a need for sleep, which means the homeostatic things have kicked in, like you need to sleep to maintain it to normal self.
So could it also affect the dependency on the circadian cycle?
Sorry, I couldn't hear the last part.
If you're sleep deprived, the need to sleep,
Yeah.
would it also affect the dependency on the circadian cycle?
That's a very good question, right? So if you're sleep deprived during a normal sleep cycle, the effect is worse as compared to when you generally don't sleep and you're not allowed to sleep. Then the effect is not that bad. So sleep deprivation only counts when you are deprived during a normal sleep cycle. So if you're sleep deprived for humans, if they're sleep deprived during the night, then this homeostatic mechanism would kick in. Yeah. Right. So let me show you the data that with which we work with. So this is recording high throughput behavioral data from larval zebrafish. I've shown you this diagram, and so you see there's a larvae in each of these wells. And let me show you a small video if that plays. So you see they are moving or not moving, and there's a red light whenever this detector detects some movement. And this experiment this was just a 10 seconds long clip, and so this experiment is done for 48 hours like I showed you, and during those 48 hours the lights are kept on. So each day is from 9 a.m. to 9 a.m. the next day, and the lights are kept on for first 14 hours and kept off for the next 10 hours, and this is repeated on the second day too. So you can see this is an activity plot. Here are the fish IDs, there are 21 fish here, and this is 2 days. And on the color axis you see the activity, which is quantized here in seconds active per minute. So let's say for example here it's 20, so they are active for 20 seconds in 60 seconds, like that. So this data is binned in minute long bins, and this is just average activity. So if you average over the fish, you see during the daytime lights on period there's a high activity, low activity during the lights off, again high and low, and this is just the distribution histogram of the activity. So there are high probability of getting 0 activity minutes, but there is a long tail of the distribution, which means there are high activity minutes too. And if I were to calculate the sleep, so this is based on traditional the accepted definition of sleep in larval zebrafish, which Shakti was talking about. So it's like if we count as 1 minute of inactivity as sleep in larval zebrafish, so they sleep around 6 to 8 minutes during the night and around 2 minutes during the day, 2 minutes per 10 minutes during the day. So you see this plot is completely reverse in pattern to the activity, because this is sleep and this is activity. Yeah, I forgot to mention this data is all from the Prober lab at Caltech, and all the data that I'll be talking about is from their lab. Now let me, sorry,
Just trying to correlate between the activity and the sleep
Yeah.
pattern. So even though they are not sleeping for the entire 10 minutes in the night,
Yeah.
why do I not see any activity in the activity heat map?
Yeah, so this is a problem of, that's why I asked the lights to turn off. They are active. So you see they are active for like a few seconds per minute, but it's not completely blue if you are asking that. They are active.
So that's somewhere between like 5 seconds per minute.
Yes, yes.
But they never reach to a minute of inactivity.
They do. So it's not that, so there are many 0 activity bins in the night, but then there are bins that have 2 seconds activity per minute or 3 seconds. So that won't be counted as sleep. Yeah. Right. So this was all about the data. Let me show you the modeling part. So what we use for modeling, like finding out sleep and wake states in larval zebrafish, is hidden Markov models. They are, I guess everyone knows about them. They are standard statistical tools, and they have been widely used for modeling sleep patterns, behavioral patterns, online speech recognition, handwriting recognition, modeling biological sequences like proteins, DNAs. So the key idea is that the observations are generated from unknown hidden states. So we don't know what's the underlying state of the system. What we know are the observations that the system emits or generates. And the second most important thing is that it makes Markov assumption on states, which means that the state of the system at time t is only dependent on state of the system at time t minus 1, but not dependent on any further history of the system. So that's the Markov assumption. So if a system here shown using a Markov chain evolves over time, so these are the observations which we can make. So at time, let's say n minus 1, the observation is xn minus 1, at n it's xn, and so on. So these observations are emitted, or let's say generated, from underlying hidden states which we don't know. So at time n minus 1 it's z n minus 1, at n is z n, and so on, and the Markov assumption is depicted by this arrow. So this state is only dependent on this, not on zn minus 2. That's what it means. And so what does the Markov model does is, if you give the data, which means if you give it the set of observations, which for our case would be those activity traces that I just showed you in the color map, it fits two sets of probabilities. One is known as transition probability, and another is known as the emission probability. So transition probability is the probability of going from one state to another in the next time step, whereas the emission probability is that given that the system is in one state, what is the probability of emitting this observation. So the model just has access to this, and it fits these two things. After it has fitted these two things, it is able to generate or predict or infer is the right word, the underlying hidden states or the sequence that the system takes, or the probability of what is the probability of state z at time t. So it can be anything out of these two. And the another very important thing in hidden Markov modeling is what is the number of states. So number of states is a free parameter. You can ask the model to fit 2 states, 3 states, 100 states, and so on. But since we are interested in assigning biological meaning to these states, these are sleep and wake states, they can't be arbitrary. So we have to first determine what are the optimal number of states that fit that data, and for that we use what is known as a Bayesian information criteria. So it weighs the accuracy of any model versus its complexity. And so if you fit the hidden Markov model with different number of states, so you fit 2 states, 3 states, 4, 5, 6, and find out what is the mean BIC. So this BIC is calculated over all the 21 fish. So you see that the BIC is minimum at 4. So the BIC minimum is always the optimal number of states in HMM. So we see that the optimum is at 4. But then there's another graph we look at, which is the distribution of optimal number of states. So each of these fish, when you fit different number of states, it can have different minimas. Where is the minima for, let's say, this fish and this fish and this fish, and then you plot that distribution. So you see that different fish might have different number of optimal states. So few fish have 3 as the optimal states, 5 or 6 or 4, but the maximum is always at the 4, or you can say the mode is always at 4. So based on just these two criteria, where is the minimum of this curve and the mode of this distribution, we take that this data has 4 as the optimal number of states. There's an interesting thing about this variability of the number of states, but we don't deal with this, at least in this project. Right. So we now know that 4 states are optimal. So we'll now fit just 4 states HMM, hidden Markov model, to our data. Now what do we call these states? So for now I am calling them as S2, S1, W1 and W2. In a minute I'll try to convince you that S2 and S1 are sleep states, and we are borrowing this terminology or nomenclature from a already published paper on Drosophila sleep or fly sleep, where they have identified hidden sleep states there. So a likely sequence, borrowing the diagram from the previous slide, could be this, like the fish goes from S2 to S1 to W1, again to S1 to W2, and these are the activities which are your like seconds per minute, how many seconds are it is active in each of these states per minute. Right. Now right? Now I've said that 4 states fit the data, but I haven't defined or characterized these states yet. We don't know the properties of these states. For that, we have to look at the parameters of the fit. So what one assumption that we make is that the emissions from each of these states follow Poisson distribution, and Poisson distribution is characterized by the scale parameter lambda. So what we find based on fitting a hidden Markov model on the data that I've shown you is that these are the 4 parameters or 4 lambdas corresponding to S2, S1, W1 and W2. So lambda S2 is almost 0, close to 0, and lambda S1 and W1 and W2 are in increasing order. Yes, so these are the lambdas for the Poisson distributions corresponding to each of the 4 states, and the Markov model can be the hidden Markov model can be captured using this transition diagram. So here these 4 are the 4 states, S2, S1, W and W2. The numbers within the boxes are the occupancies, which means the amount of time the state or the fish here spends in each of these states. So these numbers must sum to 1. The numbers here on the edges are the transition probabilities that the model fits. So if we see here, 0.87 is that the system stays in S2 in the next time step is 0.87. The system goes from S2 to S1 is 0.09. It goes from S2 to W1 is 0.03, and it goes from S2 to W2 is 0.01. So one thing you'll notice is that these numbers are progressively decreasing as it goes to farther and farther state, and it's always highest for staying within the state. And this is clearer if you look at just these green curves. So it's highest, next smaller, smaller, smallest, like that. So the mean activities associated with these states increases in this order S2, S1, W and W2. Far away transitions are less probable. Now these are the properties of the states, and I haven't yet shown you what are S1 and S2, which we are most interested in. Now for that we have to look at when do these states occur. And so here are the probabilities of these 4 states as a function of time. I don't think you can distinguish between these colors here, but this is like W2 and this is W1. So you can see that S2 and S1 have high probability of occurrence during the night as compared to during the day, whereas W1 and W2 have less probability of occurring during the night as compared to during the day. And this is just an example sequence for a given fish from the system, and you can see during the daytime, which is 9 AM to 11 PM, it mostly primarily occupies W1 and W2, but sometimes also goes to S1 and S2. But during night what you see is it's mostly in S1 and S2 states, and these are the sequences for each of the fish in this group. So you can see that based on the time of occurrence of these states, we call S1 and S2 as sleep states and W1 and W2 as wake states. But I haven't yet shown you how are S1 and S2 different from each other. They do occur at night for sure, but how are they different from each other? For that I'll show you 2 data in which sleep is perturbed, and one such data is sleep deprivation data. Sorry, you are not supposed to see this right now. Okay, so what happens in sleep deprivation experiment is for the first day, right, for first 24 hours the experiment is conducted as usual, which means 14 hours of lights on, 10 hours of lights off. But on the next day the light is kept for 14 hours on for 14 hours during the day, but it is kept on for the next 6 hours during the night and then turned off for the remaining 4 hours. So fish are forced to party essentially for the first 6 hours of night. So you can see that they have robustly higher amount of activity as compared to if you look at the same time in the baseline night or night 1. So they are more active here, which is their usual sleep period. And here's an sequence, an example sequence. So you can see that during the sleep deprivation epoch, the S2, which is generally shown here in the deep blue color, is lost, whereas it's totally occupied now by taken over by S1 state. And this is more clear if I just show you the sleep amounts during night 1 and night 2. So in night 1 you see S1 and S2 are comparable amounts, and they are high enough, 4 to 6 minutes per 10 minutes. But here if you see, this is the sleep deprivation epoch, S2 is dramatically reduced as compared to S1. But what happens post sleep deprivation is that there is a rebound in S2 sleep, and this is a hallmark of deep sleep. In mammalian sleep, it's known that if you sleep deprive a person, and if you then allow them to sleep, the deep sleep is what peaks up first. So based on this, we call S2 as deep sleep, right? And now I haven't said anything about the S1 sleep. For that, we do next experiment, which is known as tapping experiment for the lack of better word. So what we do is that we deliver a mechanical stimulus to the fish every 5 minutes throughout the night. So whenever they enter the sleep, we wake them up. Whenever they enter the sleep, we wake them up. We disturb their sleep basically. And this is just a 60-minute epoch of the data. So you can see at the taps, there's a robust increase in activities of almost all the fish. And if we fit 4-state HMM here, this is just a likely sequence of a single fish throughout the duration of experiment. I have not shown you the taps because there were too many, but what we do is we found out the response fractions, which is the fraction of fish that wake up at the tap, and we do it. So what is the fraction of fish that wake up from S2 versus what are the fraction of fish that wake up from S1? And we found that the response fraction from S1 is significantly higher than S2, which means it's harder to wake the fish from S2 as compared to S1, which indicates that S2 is a deep sleep and S1 is a light sleep. So based on these two experiments, we were able to distinguish, first of all, characterize the sleep states in terms of lambdas and their transitions, and then find out when do they occur, and then we were able to distinguish between S2 and S1. S2 is a deep sleep, S1 is a light sleep. Now, since we had this whole framework in which we are able to distinguish S1 and S2 states, we thought we should use this framework to gather more insights from sleep mutants. So sleep mutants are the animals in which there is some sleep deficit due to mutation in some of the genes. So the first group that we look at is the melatonin mutants. So melatonin mutant have a mutation in this genes NAD2, and because of that they don't have melatonin in their brain. And we all know that melatonin is important for circadian regulation of sleep, right? So these mutant animals don't have melatonin in their brain, so that's why their sleep is very less as compared to control group, which don't have any mutation or just have mutation in one copy of the gene. So I'll show you just the average activity of the control and mutants. So for controls, I'm using the heterozygous animals. So they have just mutation in one copy of the gene, which means they are still producing some melatonin, and they are essentially similar to the wild type group, which is like ++, which has no mutation. Whereas in the mutant animals, both the copies of gene are mutated. So you can see this animal group has higher activity during the night as compared to the control group, which means because of the lack of melatonin, they have high activity and hence less sleep. And that is something that the people who have worked on this data have already published. So you can see, compare the blue curve with the red curve here. So red curve shows the sleep, and you can see sleep again in minutes per 10 minutes. You can see they sleep quite a lot as compared to the mutants. So this is something what is already known in the field. So what we do is we ask what insights can HMM give us from this data, because they are just talking about the sleep as a unitary thing, as a homogeneous thing, but they are not talking about sleep states. So for that, what we do is we do HMM model fitting, but first things first, we find out what is the optimal number of states that fit the data. And we found that even though they have different dramatically reduced sleep, the number of states that fit these data are still 4 in both the groups. Now that we know that 4 states are optimal, we fit 4-state HMMs. Here are the probabilities of all the 4 states as a function of time. This is the control group and this is the mutant group. I hope you can see that during the night, if you focus, the S2 amount is very high in control as compared to the mutants, which means that the deep sleep is compromised in mutants. It's the deep sleep which is attacked here. And this is just a graph that statistically compares the 2 sleep amount, all the 4 states as a like for both the mutants and controls. So here are the controls, here are the mutants. So you can see that deep sleep has statistically reduced, whereas interestingly what we see is that the light sleep is increased, which is something the traditional methods couldn't uncover. Now next we compare the model parameters. Yes, sorry.
These states, they are not observables, but you can observe this.
No, we infer them.
Right. So
Observations are the activities, the color map that I was showing you, those are observations. But S2, S1, W and W2 are the states we infer that emit these observations.
can you briefly tell me how we infer this from the data?
Yeah, so we use hidden Markov model for that. So we fit the data and the model fits these 2 probabilities, and that's how it's able to give us the likely sequence of states. Yes.
So you have some activity.
Yes.
And then you pick the parameters.
Yes.
And you have the model
Yes.
which has these numbers.
Yeah.
So like for example in the tapping experiment,
Yes.
you have to tap in S1 and you have to tap in S2. W1 and
That's a great question. Yes.
So that I think maybe that's what you were asking.
Yes. So yeah, that's a very important point. So like so first of all, even in the tapping experiment we find 4 states is the optimal number of states, but then we see what is the state the fish is in just before the tap. So we see whether it's in S2 or S1 and see if the tap has induced some activity or not. So we infer the sequence of states. So we know irrespective of where the tap is, the model is agnostic to that, and it can infer the sequence of S2, S1, W1, W2 and so on. So I'll just show you that again. Yeah. So that's a most likely sequence for an example fish. So it goes from sometimes to S2 to S1, then sometimes to W1, and then W2 and so on.
So there's some assumption that the tabs are like well spaced out.
Yeah.
So you assume that the
No, so the model is agnostic to that. The model is agnostic to where do we put the taps, but we know where the taps are, right? So yeah.
the tab might affect the transition.
That's a very good question. So we don't look whether it goes from S1 and S2 to W1 and W2. We look whether if the system was in S1 and here do they show activity or not. So we don't rely on model prediction at the taps. We rely on model prediction in the bin before the tap.
Okay.
And then look at if it has induced some activity or not.
If you tap it too frequently,
Yeah.
probably the model would fail, right? Because
Yeah, so model's main assumption is that the data is stationary, right? And that's why we don't rely on S1 and S2 going to W1 and W2. We just check if S1 and S2 show some activity here. We go to the data space for that, for this figure. Yeah.
So even for engineering I can see some applications are very or not.
Yeah.
So, is it possible to see if a single fish has how much fraction of S1 is to S2, and then correlate that with how many times a day?
Yep.
So,
Yeah, that's possible. Yeah, but you're right. There's a lot of variability between the fish. Some animals are like I don't care if you tap me or not, I'll keep sleeping. Some animals are, you tap me or not, I'm always awake.
So does that mean the ones who are sleeping all the time, they're spending more of time in S2?
Yes, that's true. That's true. Yeah. Right. So, yeah. Yeah, so I was telling you about comparing the fits, the parameters of the control group and the mutant group. So what we observed interestingly is that the lambdas corresponding to S2 and S1 are significantly higher as compared to lambdas of the control group, which means that they are overall, the mutants are overall more active even in their sleep states. And if we look at the transition diagram, which is very interesting. So if you see, so in the red I have highlighted the parameters that are significantly different from the control group, and the arrow shows you the direction of the change. So you see that the occupancy of S2 state has significantly dropped as compared to the control group, whereas the occupancy of the light sleep S1 state has significantly increased in the mutant group as compared to the control group. Also, the transition probabilities of staying within the S2 has dropped, whereas staying within the S1 has increased. So you can now map this loss in sleep in mutants to S2 and S1 states and how these states are specifically affected. What we also see is that this far off transition, which is highly unlikely, like going from deep sleep to full awake is always highly unlikely. Person can go from deep sleep to light sleep and then to light awake and then full awake. That's generally a sequence. But this we see that these mutants have a difficulty first of all in maintaining deep sleep, but there are high chances that they go from deep sleep to full awake or the W2 state as compared to the controls. So these so we are able to map the sleep state specific changes in these mutants. So how does the loss of sleep in mutants look like in these states? The next thing, so we also look at several other mutants, for example the serotonin mutants, we look at norepinephrine mutants. So these are neurotransmitters that are very important for modulating sleep in any organisms. So and we also look at drug treatment. So for example, so this is a genetic mutation because of which there is no melatonin in the brain, but you can also get like external melatonin and make the fish sleep and then check out whether S2 is gained or S1 is gained. So one other mutant that I'll be talking to you about is known as DBH mutant, which is like, so these mutants don't have norepinephrine. So norepinephrine is important for maintaining wakefulness. So if there's no norepinephrine, you are more likely to go to sleep. Very frequently you are more likely to sleep. So this mutant is important to keep the animal awake. So let me show you the activity and the sleep plot. So you see that the mutants shown here in the red color have less activity now as compared to control, right, during the night. And here also you see they have less activity during the day too. And here is the sleep comparison. In red are the mutants, in blue, sorry, in this cyan blue they're the controls that I'm talking about. So you see the mutants sleep a lot here as compared to the melatonin mutants which slept less, right. So now we also find that 4 is the optimal number of states even for this data. Next we compare the probabilities of the 4 states. So now I think the main difference you will see here is that S2 amount has risen up in the mutants, right, during the night as compared to the controls. What we also saw, what we did not see in the melatonin case is that the wake states, right. So norepinephrine mutants have difficulty maintaining wake states. So we saw that probabilities of the 2 wake states have also dropped compared to the controls. Now I'll show you the statistical comparison. So these bar plots are for the night time comparison and this is for the day. So you can see that in mutants deep sleep has increased, light sleep has dropped, but you also see here that even during the day deep sleep has increased and the light sleep has also increased. But interestingly, we also saw that the wake 2 has decreased in these mutants. And if we compare the parameters, I'll show you all quickly. So you see that the significant differences have now shifted mostly to the wake states here. So DBH mutants have difficulty in maintaining the wake state. They have lesser occupancy of W1. They have lesser probability of maintaining the W1 state or the W2 state. They have higher deep sleep, and the probability of going from deep sleep to light sleep has also significantly decreased compared to the controls. So we were able to map out the temporal dynamics using these transition diagrams and state specific changes through hidden Markov modeling. With that, I would like to summarize. So I showed you that 4 states optimally fit the behavioral data. S2 and S1 are the sleep states. S2 is a deep sleep, S1 is a light sleep. In melatonin mutants, the amount of S2 drops and S1 rises. In norepinephrine mutants, the amount of S2 increases and W drops. The W and W2 drop. And beyond the conventional analysis, HMMs allow studying state dynamics and state specific behavior. And I'll just quickly tell you the future work, which I said in the beginning. So this will also be done in this collaboration between the 3 labs. And so next thing we want to do is map these behavioral S1 and S2 state to brain states. What do they look like in the brain? And for that we are generating these data and then modeling these data as well. So the brain data basically, and I must say that this is like a state of art technology. So using 2 photon light is very important, because 1 photon light is visible light. So 2 photon light is invisible light. So for sleep experiment, you don't want to disturb the fish while it is sleeping. So using 2 photon light is very important, and selective plane illumination. So you can divide the brain into number of planes and you can image 1 plane at a time like this. So, and the second thing we are actively doing is micro behavior tracking. So in the beginning I told you the posture is also important. So posture of sleep might be different in S2 and S1. So for that we are tracking the tail, we are tracking the eye, left eye, right eye, mouth movements and heart rate of the fish. So you can see that during lights on period the heart rate is high, whereas it dips during the lights off period. And similarly you can map the changes for other body parts. So with that I would like to just acknowledge the members in the Good Hill lab and my collaborators David, Gregorius and Ty. And if you have any feedback, please reach out to me at this email. Thank you.
So thanks, teacher, for a very interesting talk.
Okay.
So we still have time for questions.
Yes.
No.
Okay. Even further?
The
This one?
yeah.
Yeah.
So I just trying to understand the probability to be in the awake state decrease. So
Yeah.
that you will more likely, less likely to be.
Yeah, it's less likely to maintain a wake state.
But so that's why the sleeping activity in the awake state was even higher in the previous plot where you plot the sleeping activity.
You're talking about this one?
The red means the sleep.
Red?
The mutant.
Yeah.
So
Yes.
even in the day's time,
Yeah.
the activities are so average sleep is higher.
Yeah. Yeah.
So
So this is sleep, this is not activity. Okay. This is activity, this is sleep. Sorry, I've shown you both together. Yeah.
and what's
So
the
because they have difficulty in maintaining the wake state, right, they sleep less. Sorry, they sleep more, what I'm saying. Yes, they sleep more, yes. Yeah. You can see in the red curve. Yeah.
and what's the N
What is the?
in the
These N?
Yeah.
So these are number of animals.
Oh.
Can you go to the next slide?
Yeah.
Can I? So in the mutants,
Yeah.
so they can't stay in the wakefulness state, so
Yeah.
they're going to emit. Their emission probabilities to sleep will be lower. Right?
Transition properties.
So yeah, transition.
Yeah.
So it's in the mutant, the transition from W2 to S2
Yeah.
is
It will be
points.
higher actually. Because they can't maintain a wake state, they have to go somewhere else, right? So the probability of going to S1 is higher, that's why.
In the melatonin mutants,
Yeah.
the transition probability of S2 to so S2 to W2 was higher.
Yes.
They can't go.
Because it is less sleepy.
So they
can't maintain deep sleep now. They have to go somewhere else. They'll go to the wake state.
Yeah. Okay.
So they are completely opposite mutants.
I was just looking at the values and trying to.
During night,
Do they?
during night,
Yeah.
how often do you see S1 and S? I mean, the states toggling between S1 and S2.
Yeah, so that's something we have been dealing with a lot, like because we don't have any ground truth. For zebrafish, we don't know how long it should be in S1 and S2. Should we make the model more sticky, right? Whether we should somehow artificially enhance the probability of it staying in some state, right? But we don't make any such assumptions, and we see it transitions quite a lot between S1 and S2. And our theory for that is that deep sleep is like being completely unaware of the surroundings, but then this animal is a small animal and it's likely to be a prey. It goes to S1 state more likely, which is a more vigilant state. So that's the theory we came up, and we say that's why there are so many transitions between S1 and S2 throughout the night. Yeah.
These periods add up to less than 24 or more than 24 hours. So you have the 14 plus 10 hours.
Yeah. 14 and 10 hour cycle. Yeah.
So if it was, let's say, 23 or 25 hours,
Yeah.
how do you think it would?
Yeah, so we that's a very important point, and in fact there are mutants that have longer day for them. So they have 26 hours a day or 30 hours as a day. But then we try to we have entrained our fish. We have entrained the circadian cycle to 24-hour cycle by keeping lights. So since they are born or they are hatched from the egg, we keep it in 14 hours light and 10 hours dark. So we have.
Is it flexible to some extent?
Yeah, it's flexible. People work with 12 hours.
No, but it still adds up to 24 hours.
Yeah, it adds up to 24. But yeah, we as a practice, we keep it to 24 hours.
Okay, I have a question. Who is I'll do the next one. It was with the future work.
Yeah, okay.
Yes. So I noticed that, I mean, there's a clear difference between the lights being on and off for several of these, except for the first one.
Yeah, the tail
Tail
end.
line. It
Yeah.
looks like in the second on, it looks there's a lot of activity, but in the first on, there's not much activity.
Yeah. These fish are not very reliable. So one fish might show this change, but the other might not. And yeah, this is just an example.
No,
of
because
fish. Yeah.
the other left
Yeah.
and right and all of those show very clearly.
Yes. Yeah.
Oh, so this is just for one fish.
This is for one
Oh,
fish.
okay.
So we are just collecting these data and plotting to see which of the fins do they show some changes, eyes do they show some changes during sleep. And it would be great if you can map these S1 and S2 to micro behavior basically.